Hello, I'm very pleased to present to you the results about how distal cis-regulatory elements regulate tissue-specific water deficit response. This work has been inspired by the observation that climate change has a huge impact on crop yield in the field. For example, in Western Europe, the early drought of this summer have led to slowed or stopped crop development. And this has led to smaller crops, less biomass, and less grain, all three of which are huge components of yield. This is why it's very important to understand the molecular basis of this response in order to be able to identify the varieties that are most adapted to future climate conditions. In this very broad question, I'm interested in focusing on the role of distal cis-regulatory elements. Why those elements? Well, if you look at a study from Wallace and colleagues on the QTL associated to 100 different developmental traits, they found that about 2/3 of them were located outside of gene's body and potentially regulating mutation. What's very interesting is that for about half of those regulatory mutations, they were located outside of promoters in intergenic regions where the most likely functional elements are distal cis-regulatory elements such as enhancers and silencers. The issue here is that the target genes of these elements are often unknown because they are not necessarily the closest genes, they are also not necessarily the same genes depending on the cell types. That's why I'm interested into investigating the role of those very little known distal cis-regulatory elements in regulating crops' gene expression in response to water deficits. I'm interested foremost in identifying the genes and biological functions that are regulated by those elements. For that, I needed to infer the tissue and condition-specific regulatory networks. I did that using two categories of information. The first one was purely genomic and consisted into annotating the distal cis-regulatory elements with potential transcription factor binding sites. That, with the list of potential candidate target genes, allowed me to build prior gene regulatory network that summarized all of the potential interactions between transcription factors and genes. To this genomic data, we can add co-expression data obtained on different tissues and conditions. We then combine this information using Panda and Linus from the NetZoo suite. This allowed us to infer the contribution of each tissue and condition combination to the gene regulatory network. We then applied this method to maize response to water deficits. Using the model line B73 and samples from 7 tissues collected at male flowering in 2 different watering conditions, control and water deficit, we inferred the gene regulatory network for each tissue and condition combination, and we compared for each tissue the control network with the water deficit networks. The tissue for which we found the most dramatic rewiring after water deficit was for mature leaves. Here we identified 7 activated regulatory modules that were encompassing biological functions already known to respond to water deficit. It was the case, for example, for cell population proliferation that slows down in water deficit, carbohydrate biosynthesis, but also 2 kinds of response to stress, the modification of post-transcriptomic gene expression regulation, and hormonal flux. Another very interesting set of tissues were the 3 levels of internodes that all showed a very profound rewiring of energy-producing processes and also of cell wall biosynthesis. And we know that the quality of cell walls changes dramatically following water deficits. Finally, and maybe more surprisingly, we didn't find much rewiring in the reproductive organs such as the tassels, the silks, and the ear. To conclude this presentation, I'd like to let you with a few take-home messages. First, we showed that using network inference methods such as Panda and Linus from the NetZoo suite allows us to identify likely targets of distal cis-regulatory elements. Those distal cis-regulatory elements are playing an important role in rewiring the gene expression regulatory networks in response to water deficits. The extent of this network rewiring is finally strongly dependent on which tissue we're looking at. For example, leaves and internodes have a strong response, while we see no response whatsoever from hair and cells. Thank you for your attention.